Good morning. I would like to thank the organizers and the people of VLIZ in particular for inviting me to give a presentation here. As Michiel already stated, I'm more of an analytical chemist and more into public health, so it's a bit of a difficult topic for me to put some entry, but I will try to give you some insight on how I believe that analytical chemistry may aid to ensure what we call the established link between ocean and human health. So I have the advantage that I had the previous speaker already giving some insight on what aspects are important in terms of ensuring that the ocean is enabling a good human health. And from a public health point of view, I think the most important stays, and it's a bit of a narrow view of course, we have the ecological insight as well, is the food provision. There are in essence 2 billion people that are depending on the ocean for their food provision, and many others that are requiring the ocean for other aspects. One nice example is, for example, the production of pharmaceuticals. Without the coral reefs, there wouldn't have been many pharmaceuticals. For AIDS, for example, that is where the initial pharmaceuticals were found. So it helps to solve many diseases from a public health point of view. Now, in terms of the chemical pressures, there are many sources at this date that are responsible for contaminating our oceans and in essence also our seafood non-point sources. And there's a number of components that that need to be monitored. So we have agricultural practices, we have the uses of all kinds of pharmaceuticals, discharges from the municipal wastewater treatment plants, etc., but also accidents with ships that will cause the presence of all kinds of compounds, chemical residues or contaminants in our ocean. Now, in recent years, there has been a trend to measure more and more of these chemical compounds, and there has a new name been that popped up for that. We call them contaminants of emerging concern. In the past they were called POPs, but okay, names change from time to time. And in this contaminants of emerging concern, we don't only include those compounds anymore that have a persistent activity throughout the food chain and that end up in Antarctica or in our polar bears, but also those compounds that are bioactive or toxic in any way and show to be bioaccumulative and as such end up in our food supply. Now, there are many substances that are globally used and commercially sold, 30,000 more or less to be specific, but of those compounds, there are only a limited number that are measured at this point and that are included in our regulations. There have been many new regulations, there have been efforts to include as much as possible compounds, but still we have a long way to go. Because when looking at those initial analytes, the problem is that all of these compounds in the different trophic levels are metabolized. So we have many metabolites, derivatives, etc. that are not routinely measured and that may have toxic effects as well. So for which routine measurement would be important, or at least to see whether their concentrations might build up throughout the food chain. So these are what we call candidates for future regulation. Now, from a marine monitoring perspective, there are many organisms that may be used as indicator species. And as was told before, sometimes it indeed happens that some compounds, some toxins may pile up and biomagnify until the level of the shark. But using a shark or a high-end trophic chain mammal for routine measurement is not really a good strategy, and also will only reflect the contamination status at a very late point in time, which will enable us or not enable us to handle or take action quickly. The problem, of course, with all these different indicator organisms, such as mussels, which are used frequently, is that they are sessile, and as a result thereof, we need a monitoring plan, including sampling at different points. And also, to have a global impact, we need to integrate all these data. And that are disadvantages at this point, that using these indicator organisms requires, first of all, a high number of samples, only gives a view of a short period in time, and is as a result thereof not very cost efficient. And that's why we are moving towards what we call passive samplers, and I will go into that later. Now, first, something on the analytical tools and why analytical tools are, in my opinion, important to integrate in this ocean versus human health topic. So there are many instruments that are used at this point in time to look into all kinds of chemical analytes. This is an example of the apparatus that we have available in the lab. You can work with liquid chromatography, you can work with gas chromatography. It will depend on the chemical origin of the compound, although in recent years there has been a tendency towards liquid chromatography, because at this point the majority of compounds, more or less 90% is stated, can be measured with liquid chromatography, while in the past gas chromatography was more popular. Now, when moving towards alternative detection techniques and in line with having as large as possible spectrum of compounds, in recent years the high-resolution mass spectrometry has taken over from a discovery point of view at least, not a routine point of view, from the triple quads. And I will explain immediately why. Now, when looking at pollutants, contaminants, residues, you can look at one specific compound, like one fisher is capturing one fish. You have a predefined number of compounds you're looking at. You have a targeted list with the specific chemical information linked to that. And that is what is called a targeted analysis. You can have multiple targeted analysis at the same time. Going beyond a simple targeted analysis might be to have what we call a suspect analysis. You look at multiple compounds, but you don't necessarily need a standard for this, because getting standards of all, for example, the marine toxins and their metabolites will be, one, extremely expensive, and two, impossible, because there are no standards available and synthesis is not always that straightforward. So there you can make specific databases, including all the relevant chemical information on those specific compounds, and you can try to look up those compounds or see if you can find them in your extract based on their chemical information, on their mass, potential retention time prediction in terms of their chemical affinity for the column, etc. But then what we call the most holistic approach is moving forward even from suspect analysis to a truly untargeted analysis. And that implies looking at all potential compounds, metabolites, derivatives that are present in your sample. And there you don't necessarily need a database, you just have a look at the mass spectra that appear and you try to integrate them as good as possible, and specifically, most interestingly, compare two locations, two sample types towards each other. If, for example, a certain fish will have caused a disease or a direct toxic effect, you can take a blank fish or a similar fish and try to find whatever is present using multivariate statistical approaches, a bit of what we call the metabolomics strategy. But going in recent years from targeted analysis to untargeted analysis also implies a difference in methodology. While for targeted analysis, needing an individual analyte, a standard of every compound, the triple quad instrument, was mostly used. In recent years, going to these suspect and untargeted analysis, the high-resolution MS screening technology, in which you don't have any prior masses that need to be entered, you just look at everything that is present, has gained in popularity. And even the new technical advances there make that where in the past these HRMS technologies were believed to be less sensitive, in current days that is not really the case anymore. There are specific applications for which sensitivity lacks, but mostly, broadly, for the majority of compounds, it is a very good alternative technology, making sure that you can measure the entire spectrum of all compounds and their metabolites. Now, combining these high-resolution mass spectrometry-based approaches for untargeted screening and including all metabolites with a certain monitoring plan will enable you to have a good estimation of the environmental health status of potential public health risks in terms of food supply. And in this context, going from what we call the biomonitoring of indicator organisms to the passive sampler devices, for which we have now a number of projects running, is a good strategy. Because these passive samplers are in fact very simple devices. It's pieces of plastic, in fact, that are put in a sort of a cage and then can be hung at any potential interesting location. They have a more of an average weighted, time-weighted average, or give a more time-weighted average impression of what is present. They are far less time-consuming than grab samples, And you have the advantage that depending on the type of compound group you want to look at, the very hydrophobic versus more hydrophilic types of compounds, you can work with different types of materials, the more polar end capped or the very apolar types of material. And based on what is measured there, you can also make an estimation on how the bioaccumulation, etc., in a specific organism would occur. So our goal has been to integrate these biomonitoring activities in terms of specific bioindicator organisms, but also in a later phase go towards those passive samples. So I will show you now two short case studies on recent research. One was measuring the lipophilic toxins in the Belgian part of the North Sea, and another is on the endocrine disrupting compounds. So in light of the PhD project of Gabriel Orellana, who happened also in collaboration with the people of Ghent lab with Professor Janssen, we had an entire monitoring plan. Well, the first part of the work was, of course, to develop analytical methodologies enabling the detection of all these toxins, which took us more or less 2 years, which is quite a high-end application. But then, once the methodologies were in place, the goal of the project was to have a look at what specific, and in this particular case, lipophilic toxins were occurring in the Belgian part of the North Sea. And several indicator organisms from different trophic levels were measured there at several locations in the Belgian part of the North Sea, as can be seen from the map on the right. And most interestingly, it were mainly two groups of lipophilic toxins. All main groups were covered in the analysis that were discovered there, more or less in line with expectations, the aquatic acid group and a number of important metabolites, and the spirulite group. And interestingly also, but also in line with expectations, those were mostly accumulating in the shellfish, so in oyster and mussels. So from a biomagnification, but also from a bioavailability point of view, it was mainly important for future experiments in long-term exposure and activity to look into those metabolites which have not been measured or not been tested before. So if you have more questions on this, you can always ask me, but for the sake of time, I will limit this example here. And then a second project, which is currently running, has the goal to work with those passive samplers to see if they can match with grab samples on the one hand, but then also later on with potential indicator organisms in terms of screening of many different types of compounds. Steve will go into that later. But I will give you one example here for the endocrine disrupting compounds. So we have developed a method as well there to cover all the endocrine disrupting compounds, or at least as many as possible. We have 70 different compounds included there with different metabolites also. And then for those type of compounds, a specific type of passive sampler was developed, and we used the divinylbenzene passive sampler. And this in this part of the experiment, it was deployed also in the Belgian part of the North Sea for a specific time, 8 weeks here, to then perform the screening. So when working with an untargeted method, the initial goal is always to try and have a coverage of as many as different physicochemically varying compounds. So we started with these 70 EDCs for which standards were available, and of which literature had demonstrated that they are prevailing in the marine environment. And there we, in the initial screening that we did in Oostende and Zeebrugge, we had several compounds that popped up. Testosterone, for example, was one of the main prevailing compounds. But then going further from the targeted to the suspect screening, we had also a database in which more compounds were included, the ones for which no standards were available as well, or the ones for which standards were extremely expensive. And there another number of metabolites popped up, estradiol metabolites, for example. And then, interestingly, we went forward also with the untargeted screening, and based on the endocrine disrupting content of those samples, we could discriminate in between the two harbors, and further identification of compounds responsible there is currently going on. So that was the second example. Now, in terms of using high-end analytical tools, I think they are indispensable for different screening approaches, for going towards a more of a total status, or taking into account the different compounds that may occur. So HRMS proves to be a promising fingerprinting tool, and will allow, when combined with ecotoxicological research, to fingerprint more, or to show the link better with the acute toxicity and long-term toxicity in aquatic organisms, but also as such to find a causal relationship with the threat on human health that many of these compounds may have, and as such also evaluate the impact of this contamination status on the ocean health in general. So I thank you for your attention, and we will see whether there's time for questions. Thank you.